Welcome to a little bit more E&P Madness with Dr. H. Today, I want to try to dive into acid-base balance in the body. I want to try to keep this as brief as possible, but make it somewhat comprehensive for y'all. So, the very first thing to think about with acid-base balance is acids and bases, right? Hopefully, you guys already have the background to be able to understand these a little bit better, but I might as well review it for a couple of seconds. First off, what is an acid? Right? An acid is something that can donate a proton. It's a proton donor, right? In other words, an acid is a molecule that can release a hydrogen. That's what an acid is. Okay, it releases or gives up a hydrogen. Versus a base, which is a molecule that can take up or bind a hydrogen. It also releases OHs, and there's all sorts of other chemistry involved in it. But when we're dealing with acid-base balance in the human body, the easiest way to think about a base is something that will accept a hydrogen. Okay, it's a proton acceptor. So it's going to bind hydrogens. And as it binds those hydrogens, it pulls those hydrogens out of solution. As an acid releases a hydrogen, it puts hydrogens into a solution. And I find that this is a very common place for students to mistake pH and misunderstand pH, because a lot of students seem to think that pH simply means acid, and that's not correct. The pH is an inverse logarithmic log, and nobody wants to go through all that math again, I doubt. So in essence, a pH is just an inverse scale of hydrogen concentration. Hence this big ol' H right here. The P actually stands for that inverse log. So pH is relative to the acidity. This is something students have a hard time understanding. The difference between an acid and acidity. Acidity is related to the solution. Okay, acidity has to do with the solution that the acids are found in, and only if those acids have released hydrogens. So pH is related to the solution, and it's actually related to the concentration of hydrogens found in that solution. Don't forget, brackets means concentration. So concentrations of hydrogens in that solution. So when we're trying to deal with a pH, remember again, inverse, we're dealing with the concentration of hydrogens in solution. So something with a pH of 1, that actually has a very high concentration. It's going to come out to 0.1. Okay, has to do with the inverse log. Don't worry about how I got there if you don't understand it. But this number is relatively close to 1 compared to something with a pH of 14 inverse. It has to do with 1 times 10 to the minus 1 versus 1 times 10 to the minus 14. So this is going to equal 0.000000000. Okay, do I really need to go on? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 30, right? So it's a lot of zeros. So which one's bigger? Which one has more hydrogens? 0.1 or 0.0000000, right? You guys get the point, I hope. So that's what acidity is all about, is what is the concentration of hydrogens in the solution? What is the concentration of hydrogens in solution? Not what is the concentration of acid. Because acids don't have to release those hydrogens. It's only once the acid releases the hydrogen that it affects the pH of that solution. Okay, awesome. I hope that was helpful in terms of review for acids, bases, and pH. And in the human body, we have lots of acids and a few bases as well that are being released. Okay, but lots and lots of acid. So we're going to start there. And we're going to say, if we are releasing acids, then potentially those acids are entering a solution where they can dissociate, and that acid will then release a hydrogen. So what would happen to the acidity of that solution as it releases the hydrogen? The acidity would increase. Versus pH. Remember, pH is the opposite. So what would happen to the pH of that solution? The acidity increases, but the pH decreases. So try to keep that all in mind. We're going to use all that information as we talk about acid-base balance in the human body. Awesome. Really cool. Okay. Now, the next thing to think about is what do we have in the body to try to balance out acids and bases all the time? We have buffers. And buffers is the next stage of chemistry review. Okay, a buffer. So let's just define a buffer really quick. A buffer is something that attempts to stabilize the pH. That's it. It just stabilizes pH. By a buffer, depending on the buffer, can either bind up a hydrogen. Wait, what? Isn't that the definition of a base? Or a buffer can release a hydrogen. Huh. I spelt it wrong, but you get the point, right? Isn't that what we just defined an acid and a base as? And that's exactly what buffers are. Buffers aren't a brand new kind of molecule typically. They are typically something that can either bind or release a hydrogen. We have one major buffer that can bind up hydrogens in the human body. The number one buffer of the human body that can bind up a hydrogen is HCO3-. This is bicarbonate. And there's sodium bicarbonate and calcium bicarbonate, and there's a number of different types of bicarbs. Potassium bicarb drips, that sort of stuff that you can put into a patient. And bicarb has this negative charge. So when bicarb comes across a hydrogen in solution, that hydrogen will now bind to this. And when an extra hydrogen gets added to this chemical, what's it going to turn into? It's going to actually turn into the other major buffer of the human body, which is H2CO3, right? We just bound a hydrogen to this bicarb, and now we have carbonic acid. Now we have carbonic acid. Awesome. Carbonic acid and bicarb. When bicarb binds up a hydrogen, it becomes carbonic acid. When carbonic acid releases a hydrogen, it becomes bicarb, and it's just going to bounce back and forth between these two buffers. And these buffers are in the bloodstream. So what if there are too many hydrogens in the bloodstream? Well, then those hydrogens are going to be binding to bicarb. But what if there are too few hydrogens in the bloodstream? Well, then carbonic acid is going to start to release those hydrogens. I hope that started to make sense. We're going to do a couple of examples of this in just a bit. There are other buffers in the human body as well, and we'll go through some of those in just a little bit. But these are the primary buffers. How am I going to determine that? All right, these are the primary buffers, and combined, we call this the carbonic acid bicarb buffer system. Okay, this is just the carbonic acid bicarbonate buffer system. There are other buffer systems like protein buffers and hemoglobin buffers and phosphate buffers, but we're going to stick to these buffers at first here. All right, the next thing to try to think about, and we'll end up piecing all of this stuff together. You're confused about where we're going with this. Don't worry, we'll get it all pieced together. The next thing to think about is what is the normal pH of the human body? Normal pH. Not necessarily of the human body, because we don't want to really think about places like the stomach or any sort of acid-rich area. Normal pH of blood. And that's the best way to go about this, right? Normal pH of blood or the plasma is really what we're after. The normal pH is going to be a range, just like all homeostatic ranges, right? The normal pH is going to lie between 7.35 to 7.45. So that's a pH of 7.35 to 7.45. So now I start asking yourself, what do you know about the pH scale? 7 is neutral, 1 is acidic, 14 is alkaline. And here we are in the blood at 7.35 to 7.45, so we are slightly alkaline. Awesome. So what if the blood starts to drop below 7.35? What if it's 7.25? Are we acidic? Nah. But are we more acidic relative to 7.35? Yes. So anything less than 7.35 we call acidosis because it is relatively more acidic than 7.35. And what about the other way? What if we are greater than 7.45? We are going to call this alkalosis. All right, easy peasy lemon squeezy, right? Now, anything less than the 7.35 is acidosis. Anything greater than 7.45 is alkalosis. All right, so what if, I guess I should ask, what is the normal range for human life? What is the normal range for human life? Well, there's a nice easy way to be able to draw this out. Okay, just put in a 6, then put in a 7, then put in a 7.35 to 7.45, then start jumping it up. Go up to 7.8, and then you can have a final one up here at around 9. All right, so we're going to be dealing with pHs here. And if we want to say we have something that lies between 6 and 7 versus 7 to this normal range versus 7.45 to the 7.8 range versus this 9 range. So what do we know about each one of these? This is one of my favorite ways to depict this information. What if we're at 7.35 to 7.45? We're happy. Okay, it's just a nice little smiley face here, right? What if we are in acidosis between 7.0 and 7.35? I mean, this is still above neutral. We're all alkaline here, but it's acidosis because it's below 7.35. Well, this patient right here would be extremely unhappy and extremely unhealthy. And what if it was between 7.45 to 7.8? Again, we're outside of the range of healthy. This is just a nice, easy way to be able to think about this. Now, if this patient drops below 7 and is left unchecked, now we're talking a dead patient. What if it's a greater than 7.8 and is left unchecked? You got it. Okay, this is just the typical ranges. Now, can a patient drop below 7 and be revived? Sure. Okay, if this starts to drop down and down and down, we're going to start giving them bicarb drips, sodium bicarb drips, and try to figure out a way to balance out that acidosis. And if it gets greater than 7.8, we start giving them some sort of citric acid or something along those lines, something to bring their pH balance back down again. But I hope this is a nice, easy representation for y'all to be able to see. So the next thing that I'm going to do in this initial acid-base balance 1 video here is I am going to ask you guys, what do you think the normal... no, that's not the way I want to ask it. I want to ask you guys, what do you think we're typically fighting against? Do you think the body fights against acidosis more, or do you think it fights against alkalosis more? Just giving you guys a second to think about it. Acidosis or alkalosis? And the answer is it fights against acidosis more. Okay, this is why our blood tends to remain alkaline, because it's already buffered against acids. Why do we fight against acidosis more? And the answer has to do with the sources of acids. And we have a lot of different sources of excess acids in the human body. The number one source of acid, I hope most of you already have learned this, and if not, this is your time to learn it. The number one source of excess acid is carbon dioxide. Because carbon dioxide plus water, primarily in the presence of carbonic anhydrase, makes H2CO3, which is what we saw down here, right? H2CO3 is carbonic acid. And as long as carbon dioxide levels are around, we're going to have carbonic acid. And guess what? You're breathing. And as long as you're breathing and you're bringing oxygen into the system, oxygen's being converted into ATP, through metabolism and all that sort of stuff. And byproduct is going to be carbon dioxide. Did I just write CO? That's interesting. We know it's CO2. But now we're all caught up on that. There are some other sources of excess acid though, right? There are inorganic acids, and these are produced through processes like nutrient breakdown. If we start breaking down proteins, for instance, there's the disulfide bonds that are going to turn into sulfuric acid. We're going to have phosphoric acids that are produced as well. So any sort of inorganic acid is also a source of acid, obviously by the name. And amino acids and whatnot are part of now a organic. There's also an organic acid component to our bloodstream, like fatty acids, amino acids, lactic acid, ketonic acids, these keto acids, right? All of these are organic acids. And we have a lot of different sources of acids and much fewer sources of bases, of alkaline material. Okay, alkaline material, that's going to kind of be dependent on your diet, whether or not you're consuming lots of alkaline stuff. The last thing that we're going to discuss here in this first introductory video to acid-base balance, the last thing that we are going to discuss is what do changes in hydrogens, what do changes in acid and acid-base, why are they so darn dangerous? Why does the body tend to care more about acids and bases than almost anything else in the human body? Okay, it will your body will downright kill you to try to regulate acids and bases. Your pH is a very, very predetermined homeostatic measure, and your body is going to do everything it can to try to maintain homeostasis, including messing up other systems so bad that it'll kill them to try to fix it. And the reasons why, and the reasons are really deep and involved. And so for the purpose of this video, I'm not going to go into them in too much detail. But in acidosis gets up in the brain. Okay, acids are going to get up in the brain, and they're going to do things like denature enzymes. Remember, acids, hydrogens, what do? They denature proteins. Enzymes are proteins. They denature transporters and pumps and all sorts of stuff. So acidosis ends up causing CNS depression. It starts to slow that brain down. Versus alkalosis. What alkalosis is going to do is it's going to start to speed up that brain. It's going to start to cause confusion and overexcitability. A lot of this has to do with enzymatic activity. Some of it actually has to do with potassium. All right, so just one more level of review, last level of review for this particular video is that when we think of acids, we should also think of potassium, and that has to do with kidney function. So I'm going to put this in one more color here we'll go ahead and use, I don't know, this random weird color right here. I don't even know what it is. I'm not very good with colors. But what we need to be able to think about is that the DCT and collecting duct. So let's look at the kidney here. And in the kidney, the latter part of the nephron, the DCT, the collecting tubule, and the collecting duct, these have P cells and intercalated cells. In those intercalated cells, the type A intercalated cells and the type B intercalated cells, those are going to come into play. So on the DCT, and especially the collecting duct, we have a whole bunch of intercalated cells. Type A and type B. And what I want you guys to think about between these type A and type B is, what are they doing all the time? Remember, type A, type B, they're opposites of one another. They actually are opposite polarities. So type A, whatever is on the apical surface of a type A intercalated cell, it's going to be the opposite for type B. So type A, I'm just going to write it out here because this isn't part of this actual lecture. This is a review. But type A are constantly secreting hydrogens. That's one of the things they do. They're constantly secreting hydrogens. As they secrete hydrogens, they're going to be reabsorbing. And this has to do with the transporters and where they're found. But as they're secreting hydrogens, they're reabsorbing potassium. In addition, they reabsorb bicarbs. Okay, secrete hydrogens, but reabsorb potassium and reabsorb bicarbs. So let's think about this. If we have a patient that has acidosis, what is their body going to be trying to do? Their body's going to be trying to bring the acid level down. So we're going to be trying to secrete as many hydrogens as we can. But by trying to get rid of those hydrogens, what's going to start happening to potassium and bicarb levels inside of that body? They're going to start to go up. I'll bring this back into play for the next video that we watch. Type B cells just do the opposite. So that should be relatively easy. If type A cells are always secreting hydrogens, type B cells are always reabsorbing hydrogens. And what are they going to be doing to potassium? They're getting rid of that potassium. So now we're messing with potassium levels. So we're messing with resting membrane potentials. We're messing with, well, pretty much all electrical potentials, membrane potentials in the human body as we mess with that potassium. And we mess with repolarization as well. We're going to secrete potassiums, and we're going to be secreting bicarbs, and that'll come back into play for when we want to try to keep the balance. So that's the end of this particular video. I am going to be doing the next video, which now goes into how does the body start to compensate for acidosis and alkalosis? What's the body going to do specifically in order to compensate for these disorders? I hope you guys tune in to listen to it, because almost every single disease on the face of the planet will end up resulting in an acid-based disorder. You need to know this material to be effective at your job if you're in the medical field at all. All right. The end of this particular madness with Dr. H. See you in the next video.